Hey, what up guys, my name is Nobe Hernandez,
And I'm James Plain.
and today we're going to talk about aspartate aminotransferase and its role in the breakdown of amino acids within our bodies. In order to better understand aspartate aminotransferase, we must understand the overall objective of its role. Since our bodies cannot store amino acids for later use, similar to storing glucose as glycogen, our bodies breaks down amino acids into ammonium, which then gets removed through the urea cycle.
The excess amino acids go through metabolic breakdown. The amino acids go through transamination, followed by oxidative deamination, and even more. As Nobe said, the ammonium goes through the urea cycle, and then the carbon skeleton from the amino acids go through the citric cycle. For this presentation, we'll be focused on transamination and the enzyme involved.
Transamination is the process of transferring a amino group from an amino acid, in this case aspartate, to alpha-ketoglutarate to form glutamate and oxaloacetate. The reaction is catalyzed by the enzyme aspartate aminotransferase and coenzyme PLP, also known as pyridoxal phosphate, which is a derivative of vitamin B6.
The glutamate product undergoes a process called oxidative deamination, which is shown in the second reaction diagram below. So what happens is that the amine group on the glutamate is converted into an amine intermediate. NAD plus removes the highlighted hydrogen shown and also removes one of the hydrogens attached to the amine group. Another thing to note about this reaction is that the reaction is reversible, meaning that our body can take an alpha keto acid like oxaloacetate and convert it to amino acid.
However, the process of transamination is more complicated than what was shown in previous reaction diagram. The amino group does not simply detach from the aspartate and attach onto alpha-ketoglutarate to form glutamate and oxaloacetate. There are many more steps for the transformation to occur, and it is much more apparent that the enzyme and the coenzyme are involved.
Taking a closer look to the first step of the mechanism, we can see that the amine nitrogen on the lysine side chain attacks the aldehyde carbon on the PLP to form an imine. To clarify things, the lysine side chain itself is part of the enzyme Aspartate Aminotransferase. Once the imine is formed, the amino acid aspartate will attack the newly formed imine and kick out the lysine group via nucleophilic addition elimination. To get a better picture of what is happening, let's take a look at the enzyme itself.
So taking a closer look at the enzyme, we have aspartate aminotransferase here rotating around. This particular enzyme is derived from the E. coli bacteria. And inside the the transferase, we have a PLP attached to the active site, which we'll zoom in right here, right now. Zooming in into the pocket of the aminotransferase, we have PLP attached to aspartate. This compound is just an intermediate in the transamination reaction. Also, we have certain functional groups here. For example, parallel to the methyl group located here is a phosphate group. We know it's a phosphate group because there are 4 oxygen atoms attached to it. There is also a purity nitrogen within the benzene ring, within the ring, and the methyl group and the OH group are located right here. What is interesting is that the lysine group, in this case lysine 734, is close by the imine bond, suggesting this amine group was previously attached to the PLP molecule. Also, note that the lysine is part of the enzyme structure.
Now what we'll be doing is we'll be start looking at the hydrogen bonding interactions for the PLP and the aspartate on the PLP to the enzyme and other interactions. And what we'll notice is for the aspartate, you'll notice that we're having hydrogen bonding between the oxygen on the aspartate and arginine 861B, and we'll be having as well on the PLP, we'll be having hydrogen bonding with the ASN. And for the PLP as well, we are having hydrogen bonding for the serotonin right here and here with the phosphate group on the PLP, and arginine 742B with the same oxygen on the phosphate group. You'll also notice that, well, you may notice that we're having some somewhat aromatic interaction between the TRP 625B. However, it is displayed that the PLP is not an aromatic group. The possibilities that we are thinking the reason why it's not displayed as an aromatic group is that it's an intermediate at the point where it does not have aromaticity. And the intermediate we are looking at possibly would be around here, where the lysine is a positively charged, or the nitrogen on the lysine is positively charged, and that the quinonide is not aromatic. As you'll see right here, we'll show you that as aromatics is not displayed as aromatic, the PLP, and that the nitrogen on the lysine is positively charged. Going back to the transamination reaction, we see that the PLP aspartate intermediate is deprotonated to create the quininoid intermediate. The quininoid intermediate is what you see in the chimera file as the ligand. It is then protonated via hydrolysis by water and nucleophilic addition reaction to give PMP as well as oxaloacetate. To finish up the catalytic cycle, PMP is then reacts with keto acid or alpha ketoglutarate to form PLP again and glutamate.
So looking at another chimera file, we have the same enzyme but with a PMP molecule instead of a PLP molecule. And PMP stands for pyridoxamine phosphate. Right here we have PMP located on the lower left hand side of the reaction diagram, and the main difference between PMP and PLP is that PMP has the amine group from aspartate attached to the carbon, where the carbonyl used to be on the original PLP molecule. We're gonna head back to the chimera file again to take a closer look at the interactions between the enzyme and the PMP molecule. So now we're looking at the PMP molecule again, but this time we're just going to look at the hydrogen interactions that it contains. So let's start off with the phosphate group. So the phosphate group has 3 oxygens, and each of those 3 oxygens is having hydrogen bonding interactions. So like this hydrogen right here, which is the PMP O3P, is having hydrogen interactions with serine right here, as well as another serine molecule and arginine. And the other molecules are also having interactions with the serine, the arginine molecule, I mean. But there's also interactions with aspartate. For the amine group, there is also hydrogen interactions, but this time it's with the tyrosine molecule. But it only has one interaction with it, basically. We're not showing interactions with the water molecules right now because we're ignoring the hydrogen bonds for water molecules. And this is the alcohol oxygen. This alcohol hydrogen is having H interactions with the ASN. Another interesting fact about the PNP molecule is that the two ring systems are facing each other. One reason why that might be true is because the pi systems between the two rings are attracted due to having opposite polar charges or charge densities, which is why they are actually facing each other, since opposites attract.
In conclusion, aspartate aminotransferase ASPAT and it is an enzyme used to catalyze the transamination of aspartate amino group onto the alpha ketoglutarate to form glutamate. It is an important enzyme used in the metabolic breakdown of amino acids such as the aspartate, as well as the synthesis of aspartate. Without any much more to say, are there any questions?